Thank you. It's hard to get the last name right, so thank you. That was perfect. And thank you all for joining. It's really a pleasure for me to be here with you and to introduce you to Semma, and I think it's quite appropriate to follow the wonderful presentation from ViaCyte, because we too are totally laser focused on diabetes and think cell therapy is really going to be one of the things that transforms this treatment. So is there a button here to advance the slides? The green arrow. Perfect. Okay. So our vision, as you can see here, is to really change the way diabetic patients, and in particular type 1 diabetic patients, are treated and managed. And I think most of you in the audience, like most of us at Semma, are impacted in some way with a friend or a family member who's affected by this disease, and it needs no explanation what an impact that has on patients and families. And so our mission is really to use the technology that we've been developing and licensed out of Harvard to bring forward stem cell-derived islets as a therapy for type 1 and ultimately type 2 diabetes. So diabetes is really a disease in both cases of the pancreatic beta cells, which reside in the islet, a very specialized mini organ within the pancreas. And in type 1 diabetes, those beta cells, which typically respond to the glucose changes in your blood sugar when you eat a hamburger or a donut, and secrete insulin to tell your other tissues to take up that sugar and make use of it, those cells are actually destroyed by the immune system. And so in that disease, you end up in a situation where, in the absence of exogenously injected insulin, the patient can't control their own metabolism. And so there's a very clear cellular deficit in that disease. In type 2 diabetes, the complications are often a result of peripheral insulin resistance, where your peripheral tissues don't actually respond to insulin the way it should. And so that makes the beta cells over time work harder and harder and harder, and that often leads to beta cell destruction in that disease as well, and insulin dependence, insulin injection dependence in type 2 patients as well. And so our solution is really to focus on that final differentiated cell type that's missing in both of these diseases in a large fraction of patients, and that's the beta cell that resides in this specialized islet. And so the last talk talked a little bit about the process of going from a pluripotent stem cell, which has the capacity in theory to turn into any cell type into the body, into pancreatic progenitors, which we're representing here that look like a little green ball. And so our focus has really been to build on that development and on those programs, and to really make fully differentiated pancreatic islets that contain cells that are functional. So to do this whole process in a petri dish of going from a pluripotent stem cell into a functional stem cell-derived islet or a functional beta cell that secretes insulin. And so that was really the technology that we first brought into Semma and licensed from our group in Doug Melton's lab at Harvard. And we've also recognized, as I think everyone in the field of cell therapy, that how you deliver these cells really matters. The route of administration, how you protect them from destruction of the immune system. And so we are equally focused on the cell biology and cell therapy development component as on our delivery component, which I'll talk about more as I go through. So this is just a brief summary of the paper that we published a couple of years ago, and this is the technology that underpins Semma Tx. It's based on work done by Doug Melton, who's a professor at Harvard Stem Cell Institute, where I was previously with a number of colleagues, Jeff Millman and others. And it's really been Doug's life mission to transform the way type 1 diabetes is treated, and he unfortunately was very closely affected by type 1 when both of his young children were diagnosed with this disease, and he reoriented all of his academic research towards finding a cure for this disease. And we're really honored to be a part of translating that work out of the academic lab and soon into product development and clinical development. So this is an example of this type of assays we use to evaluate the cells that we make in a dish. I won't go through any of the process development of actually making the cells, but I'll just show you one of the key assays that we use, which really looks at the activity or the potency of the cells that we make in a dish. And I think it's one of the big advantages that we have of making a fully differentiated, fully functional cell product, is that it really allows you to match the assays you're using in vitro during your manufacturing development and regulatory development with the expected mechanism of action. And in this case, the mechanism of action we expect for these cells is their ability ability to sense the amount of glucose or sugar in a certain media or in the bloodstream and respond appropriately with insulin secretion. And so in this assay, you can see here, we have cells basically in a small vial that are challenged with either low glucose, which is the white bar, or high glucose, which is the dark gray bar. And as you would expect with a normal pancreatic islet, you can see higher levels of insulin secretion when the cells sense that you're getting rising levels of sugar in the media. And so we had established a proof of concept that we could do this in the lab in 2014, and have moved this into Semma. And this is work from George and Poe and other folks in our development team at Semma of really enhancing this protocol. And this was one of the examples of showing how we can improve the potency of the islets that we're actually making with this kind of approach. So we've spent a lot of time in transitioning the technology and making it robust and reproducible, but also much more pure, much more potent, much more active. And I think that's critical now as we move into our preclinical studies. So one of the other key features of any kind of pancreatic islet or beta cell therapy is not only do you want to have insulin secreted when the blood sugars are rising, but you also want to make sure that insulin secretion machinery is controlled enough so that when your blood sugar drops, that the insulin secretion machinery turns off as well. And that's one of the key safety concerns of all patients and clinicians dealing with type 1 diabetes. And so this is a slide to demonstrate that, in fact, we can actually demonstrate that working in vivo or in vitro with our cells. And so again, the white bar is exposure to low glucose for a period of time, and then sequentially exposed to high glucose and then back to low glucose. And you can see that the cells that we've made in a dish are actually able to sense and regulate their insulin secretion in response to glucose levels, which is exactly what we need them to do. This is a picture of what these actually look like. We're now doing this at larger and larger scales, which is, again, one of our key focus as we move from research to really product development. So this is large-scale production of very small, very pure islet cell clusters. You can see in green the cells. Whoops, sorry, I'll go back one. You can see in green the cells that are expressing insulin. In red is a stain for basically all the hormone-producing cells in an islet, and so those are where cells like the alpha cells and delta cells, which provide counter-regulatory signals for metabolic control, reside. And so we're working very closely on making this a clinically suitable process with clinical reagents and GMP cell lines and whatnot. So obviously, I think all of you in the audience know the ultimate goal of the scientific community is to cure mice of all diseases. And so that's, Semma is doing our part. This is an example of transplantation of those cells that you saw in vitro transplanted into mice. And what I hope you can see is that much like you saw in the last slide, there are really large numbers of beta cells, which are marked here in red, expressing insulin or C-peptide, which is a small fragment of insulin. And then in green, you're seeing cells expressing glucagon, which is, again, the key counter-regulatory hormone to insulin that really fine-tunes blood glucose control in patients. And so you can see this is out 6 months with some Semma-derived cells, and the cells are very viable and very functional here as well. So this is an example of a functional assay that we perform in vivo, again, looking to see in a fasting condition where you've just woken up, haven't had anything to eat, are you getting low levels of insulin secretion as you should? And in fact, you do. That's the blue bar. And if you then go and eat something, or in our case, inject the mice with glucose, do you see a stimulation that's related to that influx of glucose into the system? And so now the challenge for us and for everyone in the field is how do you move from that preclinical kind of studies and development into an actual product that can move into clinical development. And we've thought about this in a couple of different ways and are actually pursuing a couple of different approaches to it. I've talked to you primarily about the SC islet development work that's ongoing, and we're delighted to be working with key manufacturers to get that to the next stage. But we recognize recognize again that this delivery challenge is really, really key. And so we have an entire component of Semma, which I'll talk about in the next slide, focused on device development led by Chris Thanos and his colleagues. And so our primary goal is to move forward eventually into clinical commercial development with a Semma proprietary device and SC islet combination product into IND. We're also really interested in third-party devices and have a number of relationships with third-party devices, but I think there's tremendous challenges in the device space, and ultimately it will be a combination of innovations from our own team and externally that solve a lot of the challenges of fibrosis and vascularization and whatnot for a device-based approach. And of course, the third option, which Vaisit spoke about just before, is obviously to use this kind of cell product as a replacement for cadaveric islets. There's an enormous amount of clinical proof of concept that if you can replace these cells into patients, you can have a tremendous impact on their lives, and in the majority of cases now actually achieve insulin independence with replacement and immunosuppression. So it's a very clear potential path. The second path I want to mention just briefly is an approach that sort of circumvents that delivery challenge altogether, at least for some forms of diabetes, and that's an autologous approach that makes use of the new induced pluripotent stem cell technology, so we can actually take a sample of blood or skin from a particular patient with diabetes, turn it into what's called an induced pluripotent stem cell that then feeds directly into our manufacturing process that we've established for the allogeneic program or the off-the-shelf cell product program. So we're delighted to have the support of CERM here in California to be moving forward with that program with UCLA and Cedars and City of Hope, and focused first on a type 2 diabetic population that's very insulin sensitive. We also have a East Coast autologous program ongoing as well, focused in pancreatectomy. So we think this gives us a really unique angle into proving in the clinic that the cells are actually able to perform the type of activities that we expect them to based on, again, that clinical proof of concept from cadaveric islet transplantation. So I only have one slide on our device engineering team. We acquired a company called Cytosolve about a year ago now from out of Providence, Rhode Island, again led by Chris Thanos and Moses Goddard. And I have to say they're some of the most impressive bioengineers I've ever met. And I think they have done tremendous work in the last year with us, working really hand in hand in tight collaboration with our cell biology team to design and specifically design around the parameters that the cells need a type of device that will work for transplantation of stem cell-derived islets. And so we have a number of prototypes now, two of which are our lead candidates that really have some very innovative configurations and membranes that support the islet survival post-transplantation in ways that certainly I haven't seen before in the encapsulation field. So we're extremely excited about this, and I think hopefully next year you'll hear a lot more about where we are in that program. So this is a snapshot of our team, led by Robert Millman, our CEO and one of the founders of the company. Chris, as I mentioned, is one to highlight, who leads our delivery work and leads really the device design and transplantation studies. Walter, who's our head of manufacturing and comes out of the antibody manufacturing field and has a tremendous experience in product development and moving forward into the next phases of our development. Moses and Jason on the clinical side, both in experimental surgery and in immunology, and in endocrinology, things we know are going to be key challenges and key components of our development path going forward. And Jeff, who comes to us from Novartis and keeps the ship running in an operations role. So our scientific advisory board as well is chaired by Doug, obviously, who's the founder of Semma, and has really recruited a lot of the luminaries in the diabetes field to guide our development, and we're really grateful to have their expertise and advice, and in many cases their collaboration with Peter on the CERM grant and Gordon in the autologous program, and we've had a number of other collaborations between people in their institutions as well that's been tremendous for helping us move our product forward to the next stages of development. So in summary, I'll just say that I think we have the biological and engineering expertise to make this treatment a reality for patients, and that's really our mandate and our mission. I think to date we've really been able to develop a very efficient process of generating these types of fully differentiated islet-like clusters, transfer that process now to a commercial cell manufacturer, Lonza, who's been terrific working with us in scale-up and moving forward in our clinical manufacturing planning. We've developed, as I said, multiple device prototypes that I think are quite interesting and really give me a lot of hope that we'll be able to do something that's pretty new in the device transplantation field, first for diabetes and then potentially ultimately for other cell therapy delivery challenges as well. So we've now really transitioned completely to clinical product development. We're a preclinical stage company and eager to take the next step into treating patients, and so have initiated our combination product testing now to establish the proof of concept we need to go into INDs. And so we think about every day, I think everyone on our team thinks about what success means to us, and I think it's all driven by what patients need and want and deserve, and that's really a new option. So with that, I'll leave the last slide. Some of our key partners and supporters, we're really grateful to our investors for buying into the vision and having the audacity to hope that we can really transform diabetes. So thank you. I'll take questions outside.